Welcome to The Explainer. Today, we are taking a deep dive into Fusobacterium nucleatum. Now, this is an organism that is really shaking things up, blurring the lines between a simple oral commensal and a serious systemic pathogen. It's forcing us to rethink its role in human disease entirely. And just so you know, all the data we're looking at today comes from a comprehensive Gemini deep search, and you can find all the sources in the linked notebook.
All right, let's jump right in. Here's our game plan. We'll start with the big picture, how our whole view of this bug has changed. Then we'll get into the nuts and bolts of lab ID, how it actually causes disease, the wide range of clinical problems it's linked to, the challenges in treating it, and finally, prevention.
So, first up, this paradigm shift. This is really the core of the story. We're going to see how Fusobacterium nucleatum went from being something mostly dentists worried about to a major player for oncologists and infectious disease docs. And here it is, side by side. The change is just dramatic. We used to think of it as just a key player in dental plaque, a localized problem. But the new view? It's a formidable systemic pathogen. And even more than that, it's now considered an oncomicrobe, a microbe that's actively involved in cancer. It travels through the body, helps colorectal cancer grow, and can even make chemotherapy less effective. It's a huge shift in thinking. Okay, let's head over to the bench. How do we actually nail down the idea of this organism in the lab? Its characteristics are pretty unique, which is, super helpful for getting a diagnosis right. So, under the microscope, the number one clue is its shape. You're looking for these slender, sometimes pale staining gram-negative rods. But the dead giveaway is those pointed, tapered ends, that classic fusiform or spindle shape. It's literally where the genus gets its name. And when you try to culture it, a few things really stand out. First, it's a strict anaerobe, and it's a slow grower. You're waiting 48, maybe 72 hours. Two of the best clues for presumptive ID are that it can turn a little green when you expose it to air, and, my personal favorite, it has this brilliant chartreuse, yellow-green fluorescence under a UV woods lamp. And, critically, it won't grow on BBE agar, which is a great way to help rule out the Bacteroides fragilis group. Now, here's where it gets really interesting. Fusobacterium nucleatum isn't just one thing. It's actually a complex of at least 5 subspecies. And when you look at the genomics, the average nucleotide identity values tell us they're probably different enough to be their own species. This diversity is likely the reason they play such different roles in health and disease, and it's exactly why molecular methods are taking over. Things like 16S RNA gene sequencing or targeted qPCR assays can give you a rapid ID right from a patient sample, letting you bypass culture altogether. For a classic biochemical workup, this table lays it out beautifully. If you remember one thing, remember this. Fusobacterium nucleatum is reliably indole positive and lipase negative. That profile, combined with its morphology and culture quirks, is really the cornerstone of a definitive ID. So how does a common mouth bacterium end up causing so much trouble in places like the colon or even the placenta? Well, the answer is in its incredibly sophisticated toolkit of virulence factors. The absolute centerpiece of its pathogenic strategy is a protein called FADA adhesin. You can think of it as the bacterium's own personal skeleton key. It's unique to the invasive Fusobacterium species, and it lets them latch on to E-cadherin, a protein that's basically the glue holding our own cells together. And once it binds, it sets off this whole cascade. By messing with these cell junctions, FADA basically pries open the door between our cells. This makes the tissue more permeable, which lets the bacteria invade, slip right into the bloodstream, and then travel pretty much anywhere it wants. But wait, there's more. It has another trick up its sleeve called the FAP2 lectin. This thing is devious. It has a dual function. First, it binds to a sugar called gal-gal-NAC, which just happens to be all over colorectal cancer cells, helping it set up shop in tumors. Second, it engages the TIGIT immune checkpoint receptor on our own T cells and natural killer cells, effectively telling them to stand down. This protects not only the bacterium, but the tumor as well. So with this incredible ability to invade, spread, and manipulate our immune system, it's really no surprise that it's linked to a very diverse and pretty serious range of diseases. Yeah, the clinical impact is huge, and it connects all these different fields of medicine. It all starts with periodontitis in the mouth, but from there, its journey through the bloodstream links it directly to colorectal cancer. In obstetrics, it's a known cause of absolutely devastating outcomes like preterm birth and stillbirth. And because it can go anywhere, it can cause these deep, nasty abscesses in the liver, the brain, even in bone. Okay, so this brings us to the million-dollar question. How do we treat these infections? And honestly, therapy gets really complicated because of variable resistance and a lack of specific, solid guidelines. For the really severe stuff, like Lemierre's syndrome, you have to hit it hard. The Dutch swab guidelines, for example, recommend a combination of high-dose continuous infusion penicillin plus metronidazole. And just look at the duration, we are talking 3 to 6 weeks of IV therapy to make sure it's gone for good.
Now in kids, the principles are pretty much the same, but of course the drugs and doses are adjusted for weight. Amoxicillin clavulanate, metronidazole, and clindamycin are the mainstays, trying to find that balance between efficacy and potential side effects in a younger patient.
But here's the kicker, and this is a major headache for every clinical lab out there. EUCAST, the European Committee on Antimicrobial Susceptibility Testing, has not yet published official, species-specific clinical breakpoints for Fusobacterium nucleatum. This creates a ton of uncertainty when you're trying to interpret your susceptibility results and guide therapy.
So what do we do in the meantime? Well, the current workaround is to use the breakpoints for a close relative, Fusobacterium necrophorum, as a stand-in. But the good news is there's light at the end of the tunnel. EUCAST knows this is a gap, and they have a project underway to develop specific breakpoints for Fusobacterium nucleatum, hopefully for release in 2025. And that is going to be a huge step forward for everyone.
All right, finally, let's talk prevention. This is a tricky one, right? How do you prevent an infection from an organism that's already part of your body's normal flora? The key here is really twofold. For a patient who already has an active infection like an abscess, nothing beats aggressive surgical source control. You've got to get that pus out. But for primary prevention, trying to stop it before it starts, it all comes back to where we began, the mouth. Meticulous oral hygiene is the single best strategy to cut down the bacterial load in its home reservoir and lower the risk of it spreading.
And this whole story leaves us with a really provocative thought. Fusobacterium nucleatum is the perfect poster child for a known commensal being unmasked as a dangerous pathogen. So as our diagnostic and therapeutic tools get more and more precise, you have to wonder, what other seemingly harmless members of our microbiome are just waiting to be revealed as hidden threats, lurking in plain sight?
Thank you for joining this explainer on Fusobacterium nucleatum. If this has changed how you think about oral flora, or if you've dealt with these challenging infections yourself, please share your thoughts down in the comments. We'd love to hear from you. And of course, don't forget to subscribe for more deep dives into clinical microbiology.